Regulatory toxicology, residual analysis. Hello, friends. In this episode, we are going to learn about regulatory toxicology and residual analysis, which is a statistical method used in this discipline of toxicology. Regulatory toxicology assesses the results of toxicological investigations with regard to the potential risk to human and the environment. The existence of regulatory toxicology has resulted in establishment of a large number of standardized or core study protocols. The quality of the data generated is maintained and guaranteed by implementation of good laboratory practice (GLP) rules and certification of the relevant laboratories by the responsible authorities. Standardization and transparent documentation of the methods, animal stains, cell lines, and bacterial stains used are necessary to guarantee comparability of the data. Regulatory toxicology estimates the potential risk of exposure to xenobiotics of human or the environment. Regulatory toxicology, the rule making and compliance. The function of regulatory toxicology is to set rules and assure compliance. Regulatory toxicology is needed for product registration and allowable concentrations in food and environmental media. The purpose of scientific progress is to make this planet more and more life friendly, with ever increasing facilities and conveniences for the human beings. In this process, well being and safety of human beings as well as all other species, along with the environment of this planet, have to be looked after. Along several watchdogs, an extremely important watchdog involved in looking after the safety aspect of the process of the progress is groups of people working for the discipline of toxicology called regulatory toxicology. Their job is to set parameters so that minimum price is paid for the benefits derived from scientific progress, so that progress is made with responsibility and there is awareness about the maximum allowable damage to the human and the environment of our planet. Regulatory toxicology is an important part of toxicology and by necessity it is an interdisciplinary field. It requires integration of information from chemist, biochemist, microbiologist, cell and molecular biologist, pathologist, laboratory technicians, cancer researchers, medical doctors, animal care providers, environmentalists, engineers, mathematicians, and statisticians. Equipped with information from these sources, keeping in focus demands of the time and society, help is taken from other major disciplines of toxicology. Example, mechanistic toxicology, descriptive toxicology dealing with testing, risk assessment, and translational and clinical toxicology. After the emerge of genetic engineering in the field of biotechnology, genetically modified crops, genetically modified vegetables, genetically modified organisms, and modified drugs have dominated the markets worldwide. They are promisingly beneficial without any doubt, but they also are potentially hazardous substances for humans and environment. Since these products are genetically modified, they must be evaluated to assure that application of these products does not cause any harmful effect on humans or environment. So the field of regulatory toxicology validates the product based on technical competence and suitable data on standard reference materials and model tests. For this, regulatory toxicologist gathers and evaluates existing toxicological information to establish concentration-based standards of safe exposure. The standard safe level of a chemical is that a person can be exposed to it without causing harmful effect to health. This means regulatory toxicologists use the data provided by the descriptive toxicologist and mechanical toxicologist to decide the sufficient safe low level of drug, chemicals, or any such genetically modified product without causing harmful effects. Regulatory toxicologists are often active in government institutions. They are involved in the establishment of standards for the amount of chemical substances permitted in the environment, like in workplace or in drinking water. Regulatory toxicologists help governments to formulate regulations and put them into practice. Their advice and decisions help to minimize the risk presented by chemicals which may be hazardous to human health and environment. Majorly, regulatory toxicology deals with number one, legal aspect, which includes federal regulations, state regulations. Legal aspects include the formulation of laws, regulations, and their enforcement. For example, in the United States, enforcement falls under such government agencies as the Environmental Protection Agency, which is EPA, the Food and Drug Administration, FDA, and Occupational Safety and Health Administration, OSHA. Just like this, many other countries will have similar government agencies. In the USA, a series of laws have been created that have a basis of toxicological issues. For example, the FDA is the agency directly responsible for enforcing the federal food, drug, and cosmetic. This act includes regulations of food, drug, and cosmetic products that until then had been out of control. This law required drug manufacturers to prove scientifically that their products were safe for humans. It also prohibited the addition of poisonous substances to food products and established safe tolerance levels for such type of chemicals that were unavoidable or strictly required during manufacturing. The Federal Insecticide and Rodenticide Act, Clean Air Act, Safe Drinking Water Act, and the Toxic Substances Control Act are regulated by the Environmental Protection Agency, which is EPA. The Federal Insecticide, Fungicide and Rodenticide Act (FIFRA) regulates agricultural chemicals such as pesticides. It provides registration for requirements such as toxicological and chemical tests as well as for label information and application restrictions. The Clean Air Act includes legislation that regulates air pollutants and sets standards for air quality, including regulations on air pollution sources such as power plants, automobiles, etc. It was under this law that lead was eliminated from gasoline and emissions of sulfuric acid from industrial plants were established. The Clean Water Act enforces regulation on the production and disposal of municipal and industrial waste. Under this law, the government can set standards for emission of chemicals and in case of widespread contamination, it can recover cleanup costs and related damages from the polluting entity. The Safe Drinking Water Act relates to water intended for human consumption. Under this law, EPA can establish maximum levels of chemicals in water. Finally, Toxic Substances Control Act covers all the other chemicals produced in the USA that are not included in other acts. Under this act, the EPA can stop production of hazardous chemicals or limit their use. They may require toxicity tests to prove their safety. The Occupational Safety and Health Administration (OSHA) administers the Occupational Safety and Health Act. This act regulates health and safety in the workplace by requiring standards for worker exposure, air concentration, and industrial procedures in general. Finally, the Consumer Product Safety Commission (CPSC), through its Consumer Product Safety Act, is in charge of regulating consumer products by establishing standards for this type of products. Risk assessment. Risk refers to the probability that potential harm occurs. It was in the 70s when several US federal agencies started a systematic and scientific evaluation of the potential risk for toxicity when humans were exposed to hazardous compounds or situations. It is best defined as the determination of pathology caused by human production or activity, with the understanding that pathology is a change in some aspect of human anatomical structure or function. Risk assessment can be done either qualitatively or quantitatively. Qualitative methods checks if the material is harmful to humans under any circumstances, and it is codified by agencies, especially for cancer. Before it can be determined in any quantitative way what risk exists, it is necessary to determine whether any hazard exists. This is usually done through a systematic evaluation of the available human and animal studies. This evaluation includes epidemiological human evidence, whole animal study data on toxicology and in vitro studies. Quantitative method of risk assessment is the mathematical modeling process which yields estimates for safe or allowable chemical concentrations. This includes hazard identification. Hazard is a potential of a compound to cause damage. Dose response assessment, exposure characterization, identifying unique effects of mixtures, risk characterization, right to know and understand, safety evaluation, analytical characterization, risk assessment, remediation, and knowledge based consultancy for chemical, biological, and environment pollutants. Risk assessment is a method of systematically organizing the scientific information and its attached uncertainties for description and qualification of the health risk associated with hazardous substances, processes, actions, or events. It requires evaluation of relevant information and selection of the model to be used in drawing inferences from that information. Further, it requires explicit recognition of uncertainties and appropriate acknowledgement that alternative interpretation of the available data may be scientifically plausible. Baseline risk assessment. All risk assessment conducted by the EPA follow four basic phases. Number one, data collection and evaluation. This helps in hazard identification by collecting data of toxicological and epidemiological studies. The collected data should provide the information of adverse effect of substance with those physical or chemical properties of substance and the circumstances associated with the exposure. Number two, exposure assessment. Exposure means contact at a boundary between a human and the environment at a specific contaminant for a specified period of time. Exposure assessment identifies affected population and calculates the amount, frequency, length of the time, and route of exposure. The measured concentrations are obtained from actual samples of the source of exposure, and if the samples are not available, then the concentration are estimated based on a mathematical model. Number three, toxicity assessment. Toxicity assessment involves comparison of contaminant dose with incidence of adverse human health effect to ascertain relationship by researchers. This determines whether exposure to certain chemical causes adverse health effects. Toxicity assessment evaluates available toxicity information through databases like Integrated Risk Information System (IRIS). Number four, risk characterization. Risk characterization combines the information gathered in the exposure assessment and toxicity assessment. Through this, it quantifies risk to human health from individual chemicals and exposure pathways. This helps to evaluate cancer risk as well as non-cancer hazard separately. Risk characterization describes all assumptions and areas of uncertainty. It is not necessary that all these phases are followed in order. Each step can provide information which aids in decision making for another step. Technical and legal issues sometimes may require negotiation and gathering of new information. Risk and safety are estimated by total weight of evidence. Toxicity evidence is the basis, but often rules are modified by legal considerations as well as technical feasibility. Predictions are very important part of making regulations to protect people and the environment from the harmful effects of chemicals. Regulatory toxicologists find this out by collecting data from laboratory experiments on animals and in vitro experiments. Regulatory toxicologists conduct non-clinical and analytical studies on industrial and environmental chemicals to understand environmental monitoring, risk assessment, and remediation studies on air, water, and soil-borne domestic, industrial, and environmental pollutants. This involves mathematical models of hazard identification and remediation of water, waste, waterborne domestic, industrial, and environment pollutants. They serve as National Referral Center for Regulatory Toxicology issues. Regulatory toxicology develops new analytic methods for characterization and quantification of chemicals. This involves developing mathematical modeling of analytical method. Regulatory toxicology does toxicopathological investigations on chemicals and products. Therefore, the regulatory toxicology is involved in developing new analytical methods which helps in characterization and quantification of chemicals, develops mathematical models for analytical methods and predictive purposes, decides safe levels and does toxicological evaluation of chemical compounds and products. Residual analysis. For conducting any statistical analysis, it is important to evaluate how well the model fits the data and that the data meet the assumption of the model. For this, there are a number of different ways and variety of statistical tests to evaluate deviations from model assumptions. Residual analysis is a mathematical method to check if a regression model is a good fit. In statistics, regression analysis is a statistical process which estimates the relationship among variables. This includes many techniques from modeling and analyzing several variables when the focus is on the relationship between a dependent variable and one or more independent variables. Regression analysis helps investigator to ascertain the effect of one variable on other. If we see in the plot, it looks like the regression line is a good fit and represents the general correlation. But how do we decide that model is not a good fit? For statistical analysis, a linear regression model is not always appropriate for the data. In such cases, to assess the appropriateness of the model, residuals and residual plots are used. Residuals should not be confused with statistical errors, which is also the deviation of an observed value of an element of a statistical sample from its theoretical value. The difference between residual and error is error of an observed value is the deviation of the observed value from the true function value. Residual of an observed value is the difference between the observed value and the estimated function value. This difference is very important in regression analysis. So, why is residual analysis needed? When any model procedure is proposed, it is always needed to assess the validity of the model. This is where residual and diagnostic statistics play important role. Residual and diagnostic statistics will allow us to identify the patterns which are either poorly fit by the model or have a strong influence upon the estimated parameters or which have a high leverage. Residuals. Definition. The difference between the observed value of the dependent variable y and the predicted value y SARET is called the residual E. Each data point has one residual. Residual is equal to observed value minus predicted value. E is equal to y minus y SARET. The expected value is calculated by substituting various values of the independent variable in the developed regression equation. Both the sum and the mean of the residual are equal to 0. That is, sigma E is equal to 0 and E bar is equal to 0. Residual plots. The residual plot is a graph which is used to check if the assumption made in a regression analysis are correct. It is a graph plotted between the residuals for a particular regression model and the independent variable. Different types of residual plots can be used to check the validity of these assumptions and provide information on how to improve the model. For example, The scatter or random plot of the residuals will be disordered if the regression is good. The residuals should not show any trend, because a trend would indicate that the residuals were not independent. If residuals are put on histogram plot, they should exhibit a symmetric bell-shaped distribution, which indicates that the normality assumption is likely to be true. A residual plot is a plot or a graph which shows the residual on the vertical axis y and the independent variable on the horizontal axis x. Now, based on the dispersion of the point in residual plot, the regression model is applied. For example, if the point is a residual plot or randomly dispersed around the horizontal axis, then linear regression model is applied because it is more appropriated for the date. In other case, a non-linear model is more appropriate and thus non-linear model is applied. The table which is below on the left shows inputs and outputs from a linear regression analysis. Using this table, the residual and independent variable are used as a residual plot in the right. The values of x are 60, 70, 80, 85, 95. The values of y are 70, 65, 70, 95, 85. The values of y Saret are 65.411, 71.849, 78.288, 81.507, 87.945. The values of E are 4.589, minus 6.849, minus 8.288, 13.493, minus 2.945. In summary, now after plotting the residual plot using the data from the table, we can clearly see that the residual plot follows a fairly random pattern, where the first point in plot is positive, followed by next two points which are negative. Again, fourth point is positive, but the last point is negative. This point dispersion is clearly random, and we just established earlier that if points are randomly dispersed around the horizontal axis, then linear regression model is applied. So here also linear model provides a decent fit to the data. So linear model applied to random kind of residual plot, but which kind of residual plots need non-linear model? Following three residual plots are typical plots and gives a clear idea where to apply linear or non-linear model. As we can see, the residual plot A has all the points randomly dispersed and so the pattern is random. For such kind of data, linear model is a good fit model. But other two plots, that is B and C, has point dispersed in non-random pattern like here it shown either U-shaped or inverted U-shape. For such kind of residual plot data, non-linear model is a good fit model. If a residual plot shows an increasing trend, this suggests that the error variance increases with the independent variable, whereas a distribution which follows decreasing trend suggests that the error variance decreases with the independent variable. None of these distributions are constant variance patterns. This indicates that the assumption of constant variance is not likely to be true and the regression is not a good one. But the horizontal band pattern suggests that the variance of the residual is constant. Histogram of residuals. A histogram of residual is a good way of detecting unusual observations. It checks the assumption that the conditional distribution of y at each x value is normal. This means residual or plot on histogram to check if the variance is normally distributed. A symmetric bell-shaped histogram which is evenly distributed around 0 suggests that the normality assumption is likely to be true. If the histogram is not bell-shaped, this suggests the residuals are not normally distributed and the model's assumptions may be violated. Conclusion. As we saw in this episode, Regulatory toxicology is a discipline of toxicology which includes a team of toxicologists who gathers and evaluates toxicological data to set the sufficient safe low level of chemical substances without causing harmful effects. Regulatory toxicology deals with legal aspects for formation of laws, regulations, and their enforcements through agencies like EPA, FDA, OSHA, FIFRA, and CPSC. It also deals with risk assessment, which utilize either qualitative or quantitative route. Quantitative route includes mathematical modeling of analytical studies. The residual analysis is the statistical method involved in regulatory toxicology, which checks the regression model fit. The mathematical modeling of residual analysis includes observing the residual plot, which helps researcher to conclude if the assumptions made are valid or not. For example, the effect of different doses of chemical compound on human health for randomly distributed residual plot, the assumption that the regression model is linear is correct, and for non-random or has certain type of pattern in residual plot, the assumption is not correct. Histogram of residual checks of normality of the variance. Thank you.